It's happening right now. Creation Ministries International and Bible Discovery TV Network present the Creation Super Conference of 2011. We're blowing your mind with biblical truth in a stunning display of discoveries in science. Join us now as we go live from Muskoka Bible Camp into the Super Conference with Bible Discovery TV and Creation Ministries International. Good morning, good afternoon, good evening, wherever you are. You are looking at the beautiful environs of Muskoka Bible Center near Huntsville, Ontario. What a beautiful place to be for the Super Creation Conference of 2011. I'm Rod Hembry here with Bible Discovery TV and Quick Study Television. This morning, as we begin our series, we begin with Dr. Robert Carter, and this is going to be a fascinating discussion of genetics according to Jesus Christ. Now, Dr. Robert Carter is well studied in his field. I think you'll learn in just a moment just how well studied he is. But we encourage you to take notes, and also would like to remind you that we'll be having a break between the sessions, and just after that, we have Dr. Jonathan Safferty with the incarnation from a scientific point of view. Now, that's going to begin closer to about 10:30. And so we have a big lineup here this morning, two major speakers, and then a little bit later on tonight, Dr. Danny Faulkner with Universe by Design Part 1. He'll do Part 2 later in the week, and that will happen approximately 7 o'clock. We'll be breaking in a little bit earlier for that. So we've got three major events today at Muskoka Bible Conference. I get that right. Thank you for joining us. Now, we're getting ready to introduce, of course, this morning, Dr. Carter. So we're not going to spend any more time. We're going to go right in with Richard Fangred, who is the CEO of Creation Ministries International. Here we go. Let's learn about God and His universe.
A great day of teaching lined up here. Before I introduce our first speaker, Dr. Robert Carter from our office in Atlanta, Georgia, our U.S. office, just a few announcements. The resource area over here in the hub, if you flew here and you're finding yourself being interested in some of the books, there are some boxes that we can set up for you, and you can put things in a box throughout the week, and we will ship those things to you for free. We understand that there's all kinds of charges, of course, for extra luggage on the airplane. So if you flew here from a distance, we'll do that for you and ship your resources back home for free. Some additional things, how to structure a creation message, how to structure creation talk. I'll be doing that along with the CEO from our U.S. office, Gary Bates, and that'll be in here at 1:30. It's on your schedule, and that is basically... It's for those of you who are interested in speaking on this, or maybe you have done presentations in your church. It's going to be just an informal session. We'll probably sit down here and just have more of a chat. And we just want to share some tips from that we've learned over several decades of speaking ministry, things to look out for, things to include in a talk, how to structure a creation message. So part 1 will be today in here at 1:30, and then part 2 will be with Gary Bates and Calvin Smith on Friday at 1:30 in here. So if that sounds of interest to you, if you'd like to do a creation presentation in your own church, or if there's organizations that have approached you and you're into creation, that kind of thing, we're just going to share some thoughts on how we can help you to take your presentation to the next level. So part 1 today, part 2 on Friday. Regarding the live broadcast, I have some numbers. Yesterday for the first session in here at 9 a.m., there was 17,231 people watching, and that doesn't include the radio broadcast, that doesn't include the TV stations throughout the U.S., little community stations that are picking up the live signal and rebroadcasting it on broadcast television. And that doesn't include mobile devices like, yeah. So take the about 800 people who were here in both sessions yesterday morning, and then add 17,000 to that, and you have an idea of the audience that's being reached here this week for the Super Conference. The average viewing time was about 38 minutes. So it wasn't just people checking it out and leaving, they actually stayed for some time. About 40% of those were from Canada, 28% from the U.S., and remarkably, 13% from Australia. They were up in the middle of the night watching these things. Just incredible. We actually had a testimony from a person in Greece, in Thessalonica, where the gospel was first preached with power. And he was watching and saying, thank you so much for broadcasting these conferences. And saying, well, I'm up really late, but I'm on vacation, so I can do this. But I've mentioned it to a whole bunch of other people. Will you have these sessions available as video on demand, as like click and play, to watch later? And the answer, in case you're wondering, and for those of you watching live, the answer is yes. They will be available in the days following the conference. Next week, we'll have links up on creation.com, and also you can go to the folks that are responsible for the live streaming, BibleDiscoveryTV.com, and you can get the links there actually within we'll have the first session from tomorrow morning up fairly shortly. Is that right, Rod? Probably by lunch? Something like that? Yeah. Later this afternoon. And then as the other sessions crunch through the computer, those will be up on BibleDiscoveryTV.com as well. And then next week, we'll be sure to have the links on creation.com. For off-site guests, in case there's any guests here that aren't registered for the whole week and so on, if you're just here coming for the sessions, if the sessions are free, by the way. You don't need to pay CMI to come and listen to the speakers. You can attend every one of the sessions absolutely free. Now, the meals cost money, of course, and if you're staying here, obviously you need to pay for that. But if you're just here for the sessions and you want to use some of the grounds, you want to go down and rent a canoe or do something like that, there is an amenities fee. And you can go to the NBC registration building over here, and it's just a $5 fee, just a small fee if you want to use the facilities. But the sessions here, as I said, are completely free. Now, this morning, as we get started with our teaching, Dr. Robert Carter is with our U.S. office, and he obtained his Bachelor of Science in Biology from Georgia Institute of Technology in 1992. He then spent 4 years teaching high school biology, chemistry, physics, and electronics before going to the University of Miami to obtain his Ph.D. in marine biology. Dr. Carter was converted to Christ at an early age, but he did not know what to do with the theory of evolution. And that's a common thing for many people. Dr. Sylvester had the same kind of testimony. Came to know Christ, but then what do you do with evolution in millions of years? He always knew what he wanted to believe, but had no way to express what he was thinking and no evidence to support his views. In his freshman year in college, he was exposed to creationism for the first time and soon after adopted it. He credits Creation magazine and the Journal of Creation as enabling him to get through the in-depth evolutionary teaching of his undergraduate and graduate programs with his faith intact. Please welcome Dr. Robert Carter.
Second, please. Yeah, switching over from the one monitor to the next has made my computer not know what's going on. So here we go. Push the button. Oh, man. Tell you what. Pardon me, I am Dr. Robert Carter, and of course we're all dependent upon technology here. So I just closed PowerPoint entirely. I'm going to start PowerPoint from scratch, and then I'm going to push the button, and it's going to work this time. Okay, that's how you do it. This is going to be my first out of 3 genetics lectures for this week. This is going to be the introductory lecture. There's going to be a surprise one that's probably not on your schedule yet on Tuesday evening, which I'm sure you'll get more details about as sometime later on today or tomorrow, but I can't tell you the details because we just figured this out about, I think on Wednesday. I said, hey, Cal, I want to do another talk. He said, okay. So here it comes. That one's going to be a lot of fun. I'm going to go into a lot of details. This one is just a general talk. I need to introduce you to the topic. But because I have to use some words that some of us haven't heard in 40 or 50 years, because high school biology might have been a long time ago, or maybe it's still in the future for some of us, we need to come to some understanding that you can understand what I'm about to say. I don't want to talk over your head. So you need to be able to tell me if I said something that you don't get, and you need me to stop and back up and re-explain something. So how are we going to communicate? Well, since it's not nighttime, I can see the back of the room. Everyone practice this, right? If you do this, I will see that. Oh, good, practicing. I like that. Okay, everyone, come on, everyone practice. Now, this also means the person behind you can't see that you don't know what's going on, right? Okay, that's why I do it that way. Okay. I'm going to have to start off with a couple of words in the beginning. Like I'm going to have to use the word genome before I define genome. But I promise you, I'm going to try to define all my terms, and I'm going to define genome 2 slides from now. Fair enough? Okay. So we're talking about modern genetics. Modern genetics gives us some amazing tools. The first thing that modern genetics gave us was the ability to sequence DNA. You've heard of the Human Genome Project. That cost the U.S. government $3 billion and took over a decade. That's approximately $1 per letter in the genome. That was the Stone Age. There are companies today that can sequence the equivalent of 3 human genomes every single day. And they're talking about the imminent arrival, in fact, they've been talking about it for over a year now, the imminent arrival of the $1,000 genome. From 3 billion to 1,000 in 10 years is amazing progress in technology. In fact, the technologies that we have coming online right now, they look to be about 1,000 times faster than what we have today. So there is a coming of time, especially all of you young folks, but even some of you older people, you're going to be able to have your genome sequenced and your doctor will have it on file. And he's going to be able to make medically informed decisions based on your genetic makeup, on what medicine to give you and things like that. Now, still that's a lot of pie in the sky stuff because really $1,000 genome means nothing. It's going to cost you tens of thousands of dollars in doctor bills to have it interpreted, but that's okay. They're still talking about it. This is a picture of a genome, literally, a photograph. There's one point in the cell cycle where right before the cell divides, the chromosomes condense, and you can see them. Oh, I used to use the word chromosome. I'm about to define that also. But you can see the chromosomes in the cell. You can take a picture in a microscope, then cut out the chromosomes and line them up. This is called a karyotype. This is very important for diagnosing things like Down syndrome and things like that. It's just you look at the chromosomes, you can see them, you can see the banding. Now, looking at this photograph, let me define what a genome is. A genome is an encyclopedia. Literally. In my 3rd genetics talk, I'm going to say it's a computer program. In fact, I'm going to say it's the most advanced computer operating system that we've ever discovered, and we don't understand it, but that's my 3rd talk. The human genome is an encyclopedia. And like an encyclopedia, it comes in volumes. You have a bunch of volumes in your genomic encyclopedia. But unlike an encyclopedia, you have 2 copies of each volume inside your cells. In fact, if you strung this DNA out, this is 6 feet of DNA packed into something you can't even see. 6 feet of a string that's very sticky and very fragile, packed into a microscopic nucleus inside your cells. Amazing packaging technology. But, okay, so the genome, encyclopedia, it comes in volumes. You have 2 sets. But just like an encyclopedia, the volumes are broken down into chapters. We might call those chapters genes. You've got about 22,000 protein coding genes in your genomic encyclopedia. And those 22,000 genes produce upwards of 100,000 unique proteins. That means it's very complicated. Now, looking at this photograph, you'll notice that all the chromosomes come in pairs except for 2 chromosomes, the X and the Y on the bottom right. In fact, based on that, I know that the person from whom this picture was taken was a male. Because men have a Y chromosome, females do not. A woman has 2 X's, a man has an X and a Y. So right away we can start telling some stories about history, stories about personality, stories about what this person might look like, especially when you sequence the genome. We can say what he looked like, just a little bit about his personality, just a little bit, because your genes do influence your personality. And things like that. It's really cool stuff. But that's only one part of genetics. The second thing that modern genetics gives us is the ability to manipulate that DNA. This is a field called genetic engineering. Now, this is a terrifying field. This is very scary. We're now able in our world to ask questions we're not able to answer. Ready? What are you going to do the first time a human is cloned? What's your reaction going to be? We have the technology today to do this. The only thing preventing us is laws. But we don't have the same laws in every country in the world. And any podunk grad student in this field with maybe $10,000 worth of equipment and a willing female for a host can do this in some third world country somewhere. There are companies right now operating today in South Korea that will clone your pet for you. Little Fifi's on his last legs, send him a cell culture. They'll clone little Fifi and you get a little baby puppy back for a few tens of thousands of dollars. But, oh, Fifi's come back to life. But actually, no, it's not Fifi. It's Fifi's twin. I worked in a lab. This is from my doctoral work. I was working in a laboratory that was doing genetic engineering. I was very blessed to be there. I was working with some very smart people. And with a lot of help and a lot of wisdom from others, I was able to clone the genes from corals that cause those bright green and bright red colors. Those are proteins, very unique proteins that are fluorescent. And we cloned them into these zebrafish. And I made bright green and bright red frankenfish. And they gave me a Ph.D. for that. That's pretty cool. So yeah, I have been in this field just a little bit. And yes, I'm scared, but right now, I could give a 3-day lecture on genetic engineering. But it's not the purpose of this talk, so we're going to have to leave it for the side for some other time. That would be a fun talk. We're going to talk about the Bible. The third very important tool for modern genetics is that modern genetics gives us tools for analyzing history. Because genetics is the story of history. And the Bible, in those opening couple of chapters, claims to be a history book, does it not? It makes some very bold statements about history. Now, at this point, I'm getting a little scared again because I don't want to apply man's science to God's Word. I know the history of science. Most everything anyone has ever believed throughout world history has been wrong. A lot of what we believe today is wrong. We just haven't figured it out yet. So we have to be careful here. We don't want to take fallible science and apply it to the infallible Word of God. However, if the Bible is history, if the events of Genesis really did happen, those are events of genetics, and they should be detectable using science if we approach it carefully. Look at what Jesus said in Mark chapter 10. He said, "But from the beginning of creation, God made the male and female. Therefore, a man shall leave his father and mother and hold fast to his wife, and the two shall become one flesh. So they're no longer two, but one flesh." Wow, Jesus here is quoting straight out of the early chapters of Genesis. Did Jesus believe that Adam and Eve really lived? Luke 17, Jesus talking again. "Just as it was in the days of Noah, so it will be in the days of the Son of Man. They were eating and drinking and marrying and being given in marriage until the day when Noah entered the ark and the flood came and destroyed them all." Did Jesus believe in Noah's flood? Okay. Now, statements like this, when I was a young man wrestling with evolution, trying to figure out how evolution could fit in the Bible, I ran into stuff like this and I said, man, this is really perplexing. Because it looks, every single New Testament writer refers to the first 11 chapters of Genesis. Jesus refers to them 6 different times. I said, I don't know, man. Evolution's true, isn't it? How do you deal with all this stuff in the New Testament when they look like they believe in this young earth stuff? Well, actually, I've come around in a circle. The reason this is important, look at the way the world looks at this. This is a man named Frank Zindler, quote from Frank Zindler. He said this during a debate with William Lane Craig. He said, "The most devastating thing, though, that biology did to Christianity was the discovery of biological evolution. Now that we know that Adam and Eve never were real people, the central myth of Christianity is destroyed." Now, that underlined part, I completely disagree with. However, what happens if Adam and Eve never were real people? If there never was an Adam and Eve, there never was an original sin. If there never was an original sin, there's no need of salvation. If there's no need of salvation, there's no need of a Savior. And I submit that puts Jesus, historical or otherwise, into the ranks of the unemployed. I think that evolution is absolutely the death knell of Christianity. Now, I could have used probably a dozen different quotes from various people who draw from various non-believers who draw a direct link between the historicity of the New Testament to the historicity of the Old Testament, and says if this is full of holes like a piece of Swiss cheese, then the New Testament is full of lies. So the world is looking at this and saying, hey, Christian, you believe this stuff? This all other stuff in Genesis is a giant challenge to you. And I remember being challenged. I remember thinking things like this. What I'm showing you here is a generalized evolutionary timeline. No numbers on it, just in general. This is modeled in the out of Africa theory, which is the most common theory of human origins in the evolutionary world today. We have people living in Africa for maybe a million years called Homo erectus. And how many of them were there? Oh, hundreds of thousands, some number. But see, something happened. Something happened in world history in the out of Africa theory. Humanity nearly went extinct. We went through a population crash called a genetic bottleneck. And the entire world population was reduced to an effective population size of 10,000 people or less. And then somehow modern man evolved during this catastrophe. This is horrible. This is called inbreeding. You don't want to marry your cousin, but in a genetic bottleneck event, you have to, because there's no one else to marry. When you reduce a population size, you magnify bad mutations. I'm sure John Stanford's going to talk about that in his talk later on also. But here we have this drastic, horrible event called the genetic bottleneck, and somehow modern man evolves in here. Then our population increases in number, and then the out of Africa event happens. One line of people escapes Africa and spreads out and settles the rest of the world. Man, that's so convincing to me. But have you ever thought about the biblical model? The Bible says that we started with Adam and Eve. And that over the next 1,600 years, the Earth's population increased to some unknown number. And then we went through a genetic bottleneck where the entire world's population was reduced to 8 people. In fact, 3 reproducing couples. And from there, the population rebounded and started to expand, and then we had the Tower of Babel event where the people were spread out across the world. Have you ever thought about the commonalities of those two things? A lot of parallels. Now we're going to start talking about the Bible. There are actually 3 main events in Scripture, in Genesis, that we can address with modern genetics. Carefully, but those 3 events are the creation of Adam and Eve, the flood of Noah, and the Tower of Babel event. So let's do number 1. Genesis chapter 2 says, Then the Lord God formed man of dust from the ground, and breathed into his nostrils the breath of life, and the man became a living creature. So the Lord caused a deep sleep to fall upon the man, and while he slept, he took one of his ribs and closed up the flesh in its place. And the rib that the Lord had taken from the man, the Lord God had taken from the man, he made into a woman and brought her to the man. Oh, interesting. Did that really happen? Do you realize that those are statements about genetics? Very, very dramatic statements about genetics. In fact, the Bible says that humans had a very different history than people say in evolutionary theory. Now, let's make some predictions based on those 2 simple verses. First question, do men have one less rib than women? Sorry, I didn't want to hear the answer. Of course not. That would be the inheritance of acquired characteristics. Ribs are not passed on to children, all you people who know about the birds and the bees. In fact, if you think about it, there have been a lot of experiments in world history. If acquired characteristics are passed on, you would expect little baby girls to be born with pierced ears. So that's demonstrably false. But not only that, the rib is the one bone in the body that will regrow if it's removed properly. Our Dr. Carl Whelan, many of you know that name. Many of you actually know him. He's a director of our Australian office in Brisbane. Well, he was in a horrible car accident many years ago. And the 3rd time being rolled into the operating room for reconstructive surgery on his face, knowing they were going to take a piece of the rib out, he joked to his surgeon, there must not be much of that bone left. And the surgeon looked down and says, oh, don't you know we take out the whole thing every time? This bone will regrow if taken out properly. Very interesting. In fact, that's funny because Carl Whelan is a medical doctor and did not know that in that little bit of fact. So of course not. Now, let's ask a more modern question. Was Eve a clone of Adam? Well, think about it. God took a hunk of Adam's flesh, bone, blood vessels, cells, DNA, And he manufactured a woman from that. Well, she couldn't have been a clone, right? She's a girl. But what's the difference between men and women? It's just X and Y chromosomes. I'm not saying this did happen, but if God had taken Adam's genome and reproduced it to make Eve, left this out and just doubled that, poof, instant girl. Nearly twins. Interesting. Now I'm going to zoom up here. I'm going to just take one chromosome pair, number 2. I'm going to get rid of the rest of the genome. And I want to ask you a question. Can you see that if Adam had 2 original chromosome number 2s, and God gave those same chromosome number 2s to Eve, that there are only 2 possible variants in the human genome in the beginning? So if you're reading down the genome, A, T, C, G, C, C, A, A, C, T, T, C, 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 A, C, G, 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 oh, right here, there's an A here, and there's a T there. That means there's an A here and a T there. There's only 2 possible variants in the beginning. Can you see that? That is a genetic statement that's profound, because that puts a tremendous limit on the amount of genetic diversity we should find in the world today if we only started from 1 person. Now, if we started from 2 people, you get a whole lot more possible variation, because there's 4 possible chromosomes. If God had given Eve a brand new genome, then you could have an A, a T, a G, or a C. A lot more variation potential. Interestingly, this is absolutely what we find in genetics today. In fact, this is what we find in genetics today. There is a surprising lack of diversity amongst people across the planet. This is why they have put the bottleneck event into the out of Africa scenario. It wasn't predicted by evolutionary theory. It was predicted by creation theory. But evolutionary theory, they discovered, wow, when we look around the world, people are incredibly similar. How can that possibly be if we came from a large ancestral population that was millions of years old? They said, oh, we must have gone through a genetic bottleneck event where we lost most of that diversity. What if we started with a little bit of diversity to get the same answer? But not only that, most of the variations come in 2 flavors. Most of them. And most of that variation could be found in all world populations. Meaning, it was at Babel, it was on board the ark, and therefore it was in Adam and Eve. Let me give you an example. How many people in here have type A blood? How many in here have type B blood? Anybody in here with AB? People with type O. Oh, yes, there's many of you, of course. Do you realize that most of you with type O blood, your ancestor was an A? But you have a mutation in your gene that prevents that enzyme from being manufactured properly, so it can't stick the A antigen on the outside of the blood cell. It looks like there were 2 originals, and the third one arose through mutation. Now, actually, what I just gave you was one of the exceptions to the rule. Most of the time when we find a 3rd, a 4th, or a 5th version, those extra versions are restricted to a population. They're not worldwide. O is worldwide, meaning that mutation must have been very early in human history. But most of the genetic variation in the world comes in 2 versions. Okay. Event number 2 that we can test with genetics is Noah's flood. Genesis chapter 7 says, and Noah with his wife and his sons' wives with him went into the ark to escape the waters of the flood. A couple of chapters later, the sons of Noah who went forth from the ark were Shem, Ham, and Japheth. These 3 were the sons of Noah, and from these the people of whole earth were dispersed. Genetic statements in the Bible again. Okay, now before we go anywhere, I had to put this up one final time. And I'll need to introduce one extra piece of DNA. The mitochondrial DNA is a little circlet of DNA that's outside the nucleus. You've got a couple hundred of them in each cell. Basically, you have a cell. Inside the cell, you have a nucleus, and outside the nucleus, you have these little thing called mitochondria. They're in charge of generating energy for you. They take sugar and turn it into energy. You might have heard the phrase mitochondria are the powerhouse of the cell. You could not exist without the mitochondria. Now, interestingly, during that sugar processing, the mitochondria produce some free radicals, and free radicals destroy DNA. Why did God take this one little thing and put it outside the nucleus? Because you don't want your nuclear power plant downtown. You put it outside of town. And this is a brilliant design aspect. By taking the nuclear power plant and putting it away from the nuclear DNA, preventing those free radicals from destroying the thing that keeps us alive. In fact, one of the things that causes aging is the destruction of mitochondrial DNA over time. By the time I'm 42 years old, I know that I carry several different mitochondria within me now that I didn't have when I was born, because mutations have occurred in many of my mitochondria. And they're still chugging along, but perhaps less efficiently as they did when I was 15. And we see the long progression, the slow degradation as we age. Much of that is mutation accumulation. We can't avoid it. Okay, but in this picture here, we have two very important tools for modern genetics. We have the mitochondrial DNA and the Y chromosome. Both of these allow us to build a family tree. One of them allows us to build a family tree of all the men in the world. Obviously, because Y chromosome is only passed from father to son, right? But the mitochondria, according to theory and a lot of experimental evidence, is only passed on from the mother to the child. Men do not pass on their mitochondrial DNA, at least most always. Now, thinking about this, I apply this to my own family. I'm Robert William Carter III. We don't have very unique names in my family. I've got a cousin, actually my second cousin's son is William Thomas Carter I think 7th, if I counted it up. So my father's Robert William Carter, his father's Robert William Carter, jump over, then we have a William Thomas Carter, William Thomas Carter, William Thomas Carter. Came over from Ireland in the middle of the Irish potato famine. Came from Northern Ireland. Oh, I've got an Irish surname. Actually, I don't know what my surname is because Carter is not an Irish name. It's an English name. But the English occupied Ireland for many centuries, sadly. But during that time, a lot of people that had the last name of MacArthur changed their name to Carter. So I don't know if my Y chromosome is Celtic MacArthur or Germanic Carter. I'm going to find out one day. I want to get my Y chromosome typed so I know actually where I came from. But my mitochondrial DNA is different. See, I got it from my mother. She was a Torkelson. But that's Norwegian. She didn't get it from her father. She got it from her mother, who was a Hurlbut. That's an English name. She got it from her mother, who was a Van der Veen. That's a Dutch name. She got it from her mother, Rinstje Evert van der Vliet, who came from the northern coast of Holland. She was Frisian. The Frisians are Vikings. So I got a Viking mitochondria and a Celtic Y chromosome. You know what those people did to each other for centuries? But my children did not inherit my mitochondrial DNA. I'm a dead end. No, my children inherited the mitochondrial DNA of my wife. She's got Scotch, Irish, German, Dutch ancestry. But her mother's 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 mother, I think you can tell I'm a genealogy nerd, her whatever generation grandmother on all maternal side was rumored to be a Cherokee Indian. So my little Teutonic warriors have Native American DNA. And you would never know it. You cannot tell. In fact, genetics, there's some curveballs that we have found in genetics. Pieces of DNA that are typical of one place in the world, and yet they show up someplace where they're not supposed to be. For example, there's a family in England, an old family, and they were doing a survey of Y chromosomes in England, and all these men sent samples in, and they popped up with one that was African. So that's an African Y chromosome. And so they wrote every man with that last name in England. You can do that because it's a small country. And they said, hey, can you send us a sample? A lot of men responded. And sure enough, they all had the same Y chromosome. Then they did some genealogy, and their genealogy went back to some family in the middle of the 1700s, a white family. They said, this doesn't make any sense. There's not time for a black man for his Y chromosome to get into an English white genealogy. It's right at the dawn of the colonial era. And I tell you what, the way history has worked out, not trying to say anything about morality, it's much more likely for a man's Y chromosome to get into a native population than it is for, sorry if I use a pejorative native, but for a native Y chromosome to get into a European population. Because the white men tended to have children with the darker skinned people and not vice versa with a white woman. Just statement about history. Where did this thing come from then? Well, if you go way back in time, the Romans manned Hadrian's Wall, the border between England and that crazy area called Scotland. And they had an African legion manning that wall. And people are wondering if maybe that African Y chromosome in England goes way, way, way back. But we cannot know. And yet, the more we learn about genetics, the more we realize that racism is absolutely ridiculous. Because we share almost everything. And what separates us is just a minor component. I'm sorry, that's my next talk. All right. So, looking at this account, let's make some predictions. How many Y chromosomes were on board the ark? Aha, gotcha. Because Noah gave the same Y chromosome to all three of his children. Now there might have been some mutations in there, fine. There could be some mutations, but essentially one Y chromosome. Trick question, okay. Not a trick question. How many mitochondrial DNAs would have been on board the ark? 4. But Noah's wife is not recorded to have had any girl children. So more than likely, 3. There could have been 4. Twist my arm. Oh no, I'm not going to lose my faith. We find 4 mitochondrial lines in the world today. Okay. How many X chromosomes? That's a little trickier. More than likely 8. If by chance, there's like a 25% probability that Noah's wife gave only one, or gave the same X chromosome to all 3 of her sons, then 1 is lost and it'll be 7. And if by chance Noah had a daughter after the flood, he could have given his X chromosome to the population, and then it could be as many as 9. Don't know. But my point is, in this flood population, the people on board the ark, we can harbor a lot of genetic diversity amongst X chromosomes, a little bit of genetic diversity amongst mitochondrial DNAs, and no genetic diversity amongst Y chromosomes. Clear enough? That's a prediction of Noah's flood. Does it fit the evidence today? The answer is absolutely. Y chromosomes across the world are incredibly similar. Women, does it surprise you to learn that we really are all the same? In fact, surprisingly similar. Oh, man, oh wow. Oh, I should have had another slide right here. Last year, they resequenced the chimpanzee Y chromosome. I'm going to talk about the chimpanzee genome and the debacle of the errors that came into that later on. But they sequenced the chimpanzee Y chromosome. And they found out that the chimpanzee Y chromosome was only 70% identical to the human Y chromosome. Well, actually, it's even less than that because half of the chimpanzee Y chromosome isn't there. We've got an entire arm that chimpanzees don't have. But just comparing the stuff that we do have, 70% identical. The study said that's as much difference as they expected between man and chicken. And if you go to the common ancestor between man and chicken, the evolutionists believe that birds came from reptiles. So you have to go to the common ancestor between mammals and reptiles. That's a long time ago in their model. But if you think about it this way, they say that we're separated from chimpanzees, the common ancestor of chimpanzees, about 6 million years ago. And if you can get a 30% difference in these two strands in only 6 million years, that means that the Y chromosome must be mutating incredibly quickly, right? Why then do human males have such a similar Y chromosome? Because we must have a common ancestor, like yesterday, using evolutionary mathematics. Okay, so that's the Y chromosome. Not only that, there are, in fact, 3 main mitochondrial lineages that are found across the world. Now, the evolutionists would never say that these are the ancestral lineages. But it turns out that there are 3 main ones, and they're found on every continent. They call them M, N, and R. Now, how do we get M, N, and R? How do we get 3 lines if we only started from 1 female? Well, the answer is that because of the curse that was placed on the world after Adam and Eve rebelled, mutations are starting to accumulate in the genes. And so between Adam and Noah, the Bible lists 10 generations of men. I don't know how many generations of females there were. Today, female generation time is 10% less than the male generation time because men tend to marry women that are younger. And you take the average, it's a 10% difference. That's huge. But just going here, maybe there's 11 generations, maybe more. Between Eve and these 3 main lineages, we have some mutations that have occurred. There are 8 differences between, about 8, depends on how you count them, between M and these guys. And these 2, there's only 1 letter that separates these. Like, maybe these 2 were the Ark ladies, and maybe they were sisters. Or maybe this one was many generations removed from Eve, and these were only 10 or 11 generations removed from Eve. I don't necessarily know the answer, but we have between 4 and 8 mutations occurring in these 11 or more generations between Eve and the flood. But what about today? If I picked any 2 random people in this room, more than likely you'd be about 22, actually more than likely, on average, you'd be 22.6 mutations different from one to the other. Meaning that since these people existed, more mutations have occurred in each one of these lineages. There have only been about 150 generations since the flood. 4,500 years ago. Average human generation time today is 30 years. 4,500 divided by 30 is 150. There have only been 150 generations since we were a single family. Wow. That makes us all kissing cousins. You realize this is one of the reasons why we're so similar, because we came from a single family just a short time ago. But 150 generations, there are some people, specifically only living in Africa, that are many more times different than other people. Now, most of the Africans are in this range. But there are some people in Africa, some tribes like the San Bushmen and a few other people, that there are a whole bunch of different lines, but there are many more mutations removed. The evolutionists look at those people and say, those are the most ancient population because they have more genetic diversity. They've had more time for mutation to occur. I look at those populations and I say, oh, those poor people, they have a different population history. They've undergone more mutation because they maybe were struggling for many years and they had a smaller population size. And in a small population, you get rapid mutation fixation and rapid drift from a mean. That's later on, I'll talk about that again, et cetera, et cetera. I don't see evidence here of millions of years ago humans. I see evidence of people descending from Eve and from Noah and from Adam. Okay, I'm talking about mutations now, and I need to define what a mutation is. A mutation is literally a spelling error in your dictionary, in your encyclopedia. From one generation to the next, your encyclopedia is copied. In fact, every time one of your cells divides, your encyclopedia is copied. And it's estimated that the little molecular machines that do the copying add 1 to 3 new mutations per cell division. How many cells do you have in your body? A couple of trillion. You, right now, sitting on these benches, have every single possible mutation multiple times in your body right now. And the older you get, the more you get. It's been estimated that a 50-year-old man, or 50-year-old person, the cells lining their digestive tract, which regenerate very quickly, each one of those cells will have about 50,000 mutations that you weren't born with. But God has engineered into humanity, in fact, into most life, a brilliant design aspect. And that is that our cells that are passed on to the next generation aren't skin cells. They're not fast reproducing cells. In fact, a woman, when she is pregnant with a female, the baby, her ovaries are finished after about 30 cell divisions. Finished. And the eggs are held in that ovary in suspended animation, sometimes until 45 years later when ovulation occurs. Held there in a non-dividing state, protected, because division increases mutation load. Men, I'm sorry, it's a little bit different. When we reach puberty, our little cells start to divide, and they divide until we die. And so the older you are when you have a child, more likely you've passed on more mutations. Again, more stuff later on that. Okay, there are two different ways to measure mutations. One, I think is very straightforward. You take a family tree. You say, it's been this many generations since these men or these Y chromosomes have been separated from a common ancestor. You count up the mutations between them, and you divide by the number of generations. Very easy. When we do that, we get a very high mutation rate. Most estimates of mutation rates are 100 mutations per generation. So you were born with approximately 100 mutations that your parents were not born with. This is a very high rate. But when we hear quotes from evolutionists, they generally don't use a genealogical method, they use a phylogenetic method. They assume common ancestry. And then they'll take two genes that are shared between humans and chimpanzees, for example. They count up the number of differences and divide by 6 million, the number of years we've been separated. And that gives you a very small number. But the measurable number is orders of magnitude higher than the phylogenetic method. And I think this is indicating there's a problem with evolutionary theory. How many of you have heard that humans and chimpanzees are 99% identical? Okay, may I point out that that is a myth? And I point out by quoting a paper from 2007 by a man who's a descendant of Aaron, the priest, but that's a long story. It appeared in Science magazine, the #2 science journal in the world. He said, relative difference is the myth of 1%. And in that paper, he said, well, basically, I'm going to paraphrase, basically he said, for decades we've known that humans and chimpanzees are much more different than 1%. Then how come we keep hearing this number? Well, his conclusion, basically, he didn't really say this, but he says the term was convenient. Do you know how many Christians had their faith wrecked on the mythical rocks of 1%? Because it was convenient for the evolutionists? You cannot calculate the number. Yes, it's 1% of a certain subset of a certain subset of a certain subset of genetics. But when you add up all the rest of it, that yellow line, everything below that yellow line is stuff I pulled from other sources. I have the human and chimpanzee genomes right here on my laptop. They're right here. I ran a very simple thing. I said, wrote a little program, count up the number of letters. The chimpanzee genome, in its current state, is 13% larger than the human genome. How can we be 1% identical? There are 689 extra genes in humans that are not found in chimpanzees. That's 6.4% of our genes. Wait a minute. There have only been a few 100,000 generations since we supposedly separated from chimpanzees. It might be 6 million years, fine, but it's only a few 100,000 generations. They have to explain the evolution of entire gene families in just a few 100,000 generations. They can't do it. Natural selection can't operate that quickly. You can't get that much genetic information and that much natural selection killing off people. There'd be nothing but death all over the place just to evolve 1 or 2 of those genes, let alone almost 700. Some people estimating even more than that. There are 35 million letters that chimpanzees have an A in that spot, and we might have a G in that spot. And all people have a G, and all chimpanzees have an A, as far as we know. 35 million letter differences in just a few 100,000 generations. Natural selection can't handle that. And then I mentioned about the chimpanzee Y chromosome only being 70% identical, which would mean that it has a rapid and a radically rapid mutation rate. And yet at the same time, when we look at human males, having them all very similar, the evidence isn't there. Okay, so that's the myth of 1%. The third part of modern genetics, the third thing that modern genetics can test in the Bible is the Tower of Babel account. The Bible says this very simply in Genesis 11. Now the whole earth had one language and one speech. And it says, they said, come, let us build ourselves a city and a tower whose top is in the heavens. Let us make a name for ourselves, lest we be scattered abroad over the face of the whole earth. Okay, fair enough. But God did not like this plan because he had told them to spread out. So those first couple of generations after the flood, they're already rebelling against God. God looked down. That's not Genesis 1. It probably should be Genesis 11. Sorry about that. It says, come, let us go down and confuse their language, that they may not understand one another's speech. So the Lord scattered them abroad from there over the face of all the earth, and they ceased building the city. Essentially, God said this. Now, interestingly, looking at this, a lot of creationists have deduced that there was an ice age caused by Noah's flood, after Noah's flood. Now, the evolutionists have essentially the same scenario. Their timing is a little different. But during the ice age, ice building up in the high latitudes is going to draw the water table down. In fact, the sea level dropped in most places 200 feet or so, meaning that you could have walked from here to here, from here to Ireland, even though there's ice on Ireland, from here all the way to the southern tip of South America on dry land. In fact, you could have walked from here all the way to Tasmania on dry land, and you would only had to have crossed one open body of water. It's a deep water channel between Lombok and Bali Islands, right about there in Indonesia, but it's narrow. It would be like crossing a wide river. Cool. Some of the results of Babel? Flood legends from around the world. Now, if I was going to invent a story about a world destruction, I would probably say, oh, the world burned up in some giant forest fire, or something like that, or maybe a big dragon came and ate everything or something. I would make it up. But it turns out that people across the world share a common story of a giant flood that that cover the world. We have genealogies of various peoples going back to biblical figures. In the book After the Flood by Bill Cooper, he tracks most of the pagan royal houses of Europe go back to Japheth, pre-Christ, pre-Christianity. The Miao people of one of the minority peoples in China, they have a genealogy that goes back to Noah through Japheth, pre-Christian contact. Interesting. There's all sorts of cultural connections, like why did people build big pyramids in ancient times across the world? I think it's because we came from a single population. Looking at Genesis 9 and 10, this is called the Table of Nations. In Genesis 9 and 10, there are 16 sons that are named to Shem, Ham, and Japheth. Now, if you assume that, well, okay, there's going to be about 16 daughters also. So in Shem's generation, there are 6 people. That means in the next generation, there are about 30 people. At that rate of growth, there will be about 150 people in the next generation, about 750 in the 4th generation, and about, 3,000 or so people. Now, the numbers get iffier as you go farther on, but about 3,000 people or so in the 5th generation. In Peleg's day, in his day the earth was divided, the Bible says. And if you look at the Peleg account, Tower of Babel, Peleg, in his day the earth was divided, Tower of Babel account. He is in the middle of the Tower of Babel account. There are some creationists who believe that the division of the earth was a separation of the land masses. I have big problems with that, specifically the world catastrophes that would have had to have occurred after the flood that aren't mentioned anywhere in history. So I do not like that. I do believe that Peleg is the earth was divided according to language in his day. But just looking at that, I don't know how many people are at the Tower of Babel. So let's just round this off to somewhere between 1,000 people and 10,000 people. Now that's interesting because that's the evolutionist number for the Outer Africa event, an effective population size of 10,000 people or less. Now these are 2 totally different numbers, came up with 2 totally different methods. I think it's just curious that they happen to line up, not that they actually, there's a reality behind that. Now if we look at this, here's a Tower of Babel, that funny trapezoid in the middle, there's a bunch of people. How large was the Babel population? Now take that population, take 50 people out of there and send them east. Take 50 people and send them west. Take 50 people and send them north. Can you see that most of the populations are going to get most of the colors in them? The colors represent genes. If the population is large enough and you divide it into large enough subpopulations, then each of the subpopulations is going to get a pretty good representation of all the genes at Babel. And so if you took a people group, think people from Iceland, and put all their genetic diversity in there. And then take another people group, maybe people from Fiji or Tonga. Put them in there with all their genetic diversity. Now take another group of people. Pick somebody. I don't know, I'm running out of people. Native Americans. And put them in there. And what we see is that, well, first of all, most of what we share more than 99% of everything. And what separates us is just a fraction. Louis Quintana Murci, famous population geneticist, he said this recently. He said, but the genes that explain the phenotypic differences, that means the way we look. The genes that explain the way we look between populations only represent a tiny part of our genome, confirming once again the concept of race from a genetic standpoint has been abolished. My friends, if we all came from Adam and Eve, and if we all came from Noah and his family just 150 generations ago, there's no basis for racism whatsoever. And now modern genetics is learning that this is true. If we look at just one type of diversity, these are letter differences between people. Just looking at letter differences. Here's Europeans, Asians, and Africans. You can see that the bulk of the letters, like everyone in the world has A's and G's at some location. Everyone in the world has C's and T's in another location. But there are a few places where the Europeans have a unique letter at maybe chromosome 15, position number 6 million. Some Europeans have a C and no one else in the world has that. Well, the Asians have a few unique ones and the Africans have many unique ones. This is the reason why the out of Africa theory came about. They said Africans have more genetic diversity. In fact, you take 2 people from neighboring tribes, neighboring people groups in Africa. They can have more diversity, more differences between them than a person from Iceland can have from a person from Japan. Europe is essentially the equivalent of one African tribe. That's how much genetic diversity is within that continent. It is amazing. But evolutionists say it's because of age. I don't believe so. There's one thing, however, that are not shared amongst people. What is not shared are deletions. See, God engineered into your genome 2 copies of your genome. It's a brilliant design. If it wasn't for that, you would already be dead. Humanity would already be extinct. But we have 2 copies, so when one copy breaks, maybe the copy from dad, well, the copy from mom is still good, so you can still live. Each one of you sitting here right now is probably missing over 1 million base pairs of DNA from your genome in different pieces that have been deleted accidentally throughout human history. Now, interestingly, those deletions are not shared amongst populations. Most of the diversity is shared amongst population, but the deletions are not, meaning they're new, meaning they're post-Babel, and meaning the human genome is falling apart. It's not evolving upwards, it's evolving downwards. And we are going to go extinct because of this accumulation of mutations over time. But we're not going to go extinct because Jesus promised that he's going to return and there will be people alive on earth when he returns. Here's another study. I said, the human genome. Well, here's a study that analyzed 20 human genomes. And they concluded we summarized different categories of variation in each genome. In total, across all 20 of the genomes, finding a surprising number of variants predicted to reduce or remove the proteins encoded by many different genes. So we have even more evidence that people today are missing genes. In fact, surprising for them because you're not supposed to delete information over time. You're supposed to evolve information over time. But what we see is things falling out of the genome rapidly. And evolutionists did not predict that. Genesis chapter 10 says something really interesting. It says, now these are the clans of the sons of Noah according to their genealogies in their nations. And from these, the nations spread abroad on the earth after the flood. That right there actually is a genetic statement. And it is the key to understanding modern genetics. Because when God separated the population according to language, he divided the population according to male ancestry. And male ancestry means Y chromosome. He used language to separate the Y chromosomes of the world and spread them out. But if you think about the early flood, there's only 3 families, Shem, Ham, and Japheth. Their 3 wives. Would you expect the daughters of Shem to be marrying the sons of Ham and the daughters of Ham to be marrying the sons of Japheth? Well, yeah, there's no one else to marry. There's one language, there's one culture, there's one family. There should be interbreeding. Interbreeding, which means all those lineages are all scrambled up until Babel, when God sorts them again, and he sends them out according to Y chromosome. What happens to the mitochondrial DNAs? They're not sorted, they're scrambled, which is why we find those 3 main mitochondrial DNAs everywhere. But we don't find the Y chromosomes everywhere. Because when we look around the world, there is some variation amongst Y chromosomes, and they're very typical variations, which is why I said earlier, a typical African Y chromosome was found in England. Well, there are typical things that this is typical of this region, this is typical of this region, because after the clans got to where they were going, mutations occurred in those Y chromosome lineages. And so each area of the world has specific Y chromosomes. That's not true of mitochondrial DNA because they were randomized in there. But now, wait a minute, think about this. Who in history moves around more? Men or women? Men! Who goes off to fight wars? What do men do when they conquer the city? Don't say it out loud. Who is rumored to have a woman in every port? Sailors. Who's the philanderer and who's the person who stays at home pining away after her wandering husband? How is it then that the Y chromosomes have stayed put and the mitochondrial lineages have spread out? Oh. This is the opposite of the prediction of human nature. The reason is that after Babel, God put a genetic signal in the population that despite human nature, that signal has not been erased. And also because it has not been tens of thousands of years. It's only been a few thousand years. Or that would not be true any longer. This is a evolutionary model of the out of Africa theory. I pulled this from this website, mitomop.org. They're charting mitochondrial lineages, female lineages across the world. So they have somewhere in deep, dark Africa. Sorry, I don't believe in deepest, darkest Africa, but I know. They have somewhere in Africa, they have humans for a long time evolving, and then these three main lineages evolve, L1, L2, L3. There are other ones. And then L3, for some reason, some more mutations occur in here, and we get three main lines, M, N, and R. And only a subset of the L3 lineages, only these guys escape, and they go across the world. Now think about this. They're saying that there's been a recent migration of people traveling through the Middle East with three main mitochondrial lineages, moving into previously uncharted or uninhabited territory in the recent past, in small populations. Every single one of those points comes out of the Tower of Babel account. This didn't have to be true. Pick all the creation stories across the world. What's the likelihood that the actual data would be a nice fit to the biblical model? It would be very unlikely. And yet, do you know why they had the starting point here? It's because of genetic diversity. It has to be older. But the genetic diversity in this region is not much less than this region. In fact, diversity in Africa is here. Diversity goes down as you go this way. It goes down as you go this way. Diversity goes down as you go this way. It goes down as you spread out from some circle in this region here. So putting that starting point in Africa actually is a bit of a bias. You can't actually do it. It's statistically likely or whatever, but it's very easy actually to shift it over here. But why would the population geneticists not do that? Because then it becomes a Tower of Babel story, and that's not tolerable. I have a paper in the Journal of Creation. You can Google, on Journal of Creation, go on the search box, type in Carter space Africa, and this paper will pop up. I looked at all the assumptions behind the out of Africa theory, and then I looked up papers that discount every single one of those assumptions. If any one of these assumptions fails, the out of Africa theory is dead. They depend upon what's called a molecular clock. It depends upon mutations occurring at the same rate in all populations across all of time, which actually is a ridiculous proposition. Because we know that people carry defective DNA enzymes, DNA repair enzymes, and those lines accumulate mutations faster than others. We know that environment influences mutation accumulation. We know that population size and population history and average age of marriage and the family size, all these different things influence, the demographics influence the accumulation of mutations over time. They're assuming that every population in the world has exactly the same history, which is untenable. And they refuse to allow any mutation that affects the speed of mutation accumulation. In other words, you can't have a mutation in a DNA repair enzyme. You can't have this people over here have one style of DNA repair enzyme and this one over here have a different style. Like maybe this one's more likely to replace it with an A and this one's more likely to replace it with a G. That's not allowed. There are so many things behind this that are just treading on air. In my other talks, I'm going to get into this in more depth and throw you some charts and numbers that are going to knock your socks off. Okay, let's throw some conclusions here. First of all, modern genetics has essentially validated the big picture of genetics. It has discovered evidence for the creation account, the flood, and the Tower of Babel. It's written in our genes right now. Now, as a scientist, I'm actually very skeptical. I don't believe the science can prove hardly anything. Honestly, you cannot prove to me that yesterday existed. You can't do it. Now, I believe yesterday existed from experience, but that's experience, not science. Anyway, as I said earlier, a lot of what we believe in science is going to be wrong, et cetera, et cetera. Science is a negative proposition. We weed out possibilities. It's very difficult to say, absolutely, this is actually the case. But you can weed out possibilities. And the Bible has not been weeded out. Despite the best evidence, the best attempts of the evolutionists to discount the Bible, it's still standing. And it still fits the data. And that, to me, is incredibly exciting. Now, if you want to learn more, this talk, I gave the same talk, or a similar talk to this one, at the Creation Super Conference in Australia 2 years ago, I think. It's called Mitochondrial Eve and the Three Daughters of Noah. Essentially the same thing. If you want to hear it again, you want to share it with someone, that DVD is available on CMI's website, and it's available on our book tables out here. John Sanford is going to give a talk on genetic entropy later on. And I want to promo, in case he forgets, I want to promo his book, Genetic Entropy and the Mystery of the Genome. A brilliant exposition from a person who was an evolutionary geneticist for many years. When he became a Christian and a young earth creationist, he writes a book that is an insider's look at the mathematics of evolutionary theory. And he shows conclusively, using the evolutionist's own words, that the emperor has no clothes. Fantastic book. And if you really want to cut deep, I will tell you right now that creationism is undergoing a revolution in genetics. And we are building up some unbelievably interesting ideas, and the cutting edge of these ideas are appearing in the Journal of Creation. If you don't subscribe to the Journal of Creation, most of you in here are creation junkies. That's why you're here. You should be subscribing to the Journal of Creation. I have been unbelievably blessed by some of the things I've read in here. I wouldn't be standing here right now if I hadn't been reading this for the last 15 years. Because it has trained me to rethink and has given me all sorts of ammunition I did not have before. Now, for all the people out watching on the internet, of course you know about our website. But don't forget, we also have a Twitter and a YouTube account. And we're all over the internet. There's many places we can go. All right, thank you very much. Oh, wait a minute. Don't say anything. Two things that I don't have slides for. About on the tables, we've got really cool packs. We don't have these in America. I'm going to go back to America. In fact, my boss is here. I'm going to say, hey, boss, we need to do this. This is the DVD Discovery Pack. 1, 2, 3, 4, 5, 6 different topics on a range of issues, which will be a great introduction to basic creationism, to hit all the high topics. Now, some of you in here aren't very familiar with creationism. Something like this would be a great starter. If you've got someone that you want to share this with, that might be a good thing. Or this one, the Main Issues Packs. And we have The Greatest Hoax on Earth, Creation, Facts of Life, and Genesis and the Gospel Connection DVD. A great place to start. Now, after that, there's tons of stuff where you can really dig deeply, and I'll leave that up to you. Okay, thank you very much. We're going to open up the floor for questions for about 10 minutes. Okay, stump the chump time. Sir.
Given the fact that we come from common parents, how does genetics explain skin color variations? Is that possible that Noah's sons could?
Being that we came from the same parents, how does that explain skin color variations? Is it possible that Noah's sons may have been a different color? Okay, great question. First of all, skin color variation. I am European. I have a mutation in my M1CR gene that prevents the accumulation of melanin in my skin. If you are from Asia, I do believe that the latest data says that Asian people who are light-skinned have a different variant of that gene that causes a different reason for them to accumulate less melanin in the skin. There are some people on the subcontinent of India, Australia, and Africa who are as dark as you can imagine. I believe it's because that gene is broken into the on position and can't be turned off. So they just produce tons and tons of melanin. But it's the same gene. I do believe that probably Adam, Eve, Noah, and his children were middle tone brown. And that since then, other things have occurred. Now, it might have been that this is in early variation. I don't necessarily know. But being that light-colored skin is not spread across the world, it looks to me like it's recent variation, recent mutation. There are some people who believe that Ham had dark skin. This is false. There are some people who believe that when God cursed Ham, therefore all Ham's descendants are dark, and therefore that's why Africans are dark. This is not true. I just mentioned other world populations that are not from Ham that are also dark. But also God cursed Ham's son Canaan, not Ham himself. And Canaan lived in Canaan land, which is in the Middle East, which is now Israel. And had nothing to do with skin color as far as we know, et cetera, et cetera. There's actually, I believe in the Creation Answers book, that is addressed. I don't know in what chapter. Someone knows it better than me. I'm sure that question is here in the Creation Answers book. So, great question. More information on our website. Sir?
I used to hear that the theory that the Aboriginals in Australia were of an entirely different line from the whole rest of the world. I'm curious where that theory went and how it fits with your work.
The question is about Aboriginal Australians and the theory that they were from a totally different line. I'm not totally up to speed on Aboriginal Australians. I've read different things, and in fact, I think the reason I'm not up to speed is because there's an evolutionist war going on between different camps and what they say, who came from whom. Darwin wrote about the various groups of people in the world, and he divided up people into multiple different races. He tried to make them species, but he was a little vague on whether they were species or not. But he had the Aboriginal Australians as one group of people, almost like monkeys. He had them at the bottom of the evolutionary ladder, as close to monkeys as possible without being a monkey, essentially. That's kind of stuck in evolutionary theory and thinking for a while. But it's clear that Aboriginals are descended from people living in Asia. They're a subset of Asian people. Now, people think they got there a very, very long time ago. There are some people who say that some of the subpopulations in India are the source population for the native Australians, but that's very contentious. People get very angry when you say that. But they came from the Northwest as they migrated down. The timing depends upon mutation count and depends upon mutation rate. I don't think rates are constant, therefore I don't believe the timing based on just strict mutation count. Boy, that was a convoluted answer. I hope I did that justice. Sir in the back. Oh, it's my boss. Yes, sir.
Boss, but anyway, just going to add something being an Aussie. Rob, you might not be aware, Don Batten, one of our scientists in Australia, has actually shown some of the latest research shows that two major population centres for Aboriginals. We have Western Desert Aboriginals, which is the west coast where I'm from, and on the eastern side actually probably came down from two different regions. So Asia, as you said, and India, the subcontinent, and the East Coast Aboriginals actually look like they came from some of the Melanesian populations and the islanders and from Southeast Asia. So actually it might not all be descended from one particular group.
Thank you. I had read that and forgot. Gary Bates, back there, one of our speakers, a good friend of mine, and my boss, he just reminded me that another one of our speakers, Dr. Don Batten, has done some studies on Neanderthals, and I guess he's using other people's data who have said that looks like the Neanderthals, Aboriginal Australians. No, that was a slip of the tongue. That was rude of me. I did not mean that. The Aboriginal Australians might have come from two ancestral stocks, where the Westerners might have come from Asia and the Easterners might have come from Melanesia, based on their genetics. Neat stuff, but again, it's a subject I haven't delved into it as much as I need to. Ma'am? Yes. DNA replication is the question is, I just said that we get mutations from our parents because of DNA replication, but on the other hand, DNA replication is incredibly accurate. How do those two facts mesh? DNA replication is amazingly accurate. These little teeny machines can duplicate 6 feet of DNA in a matter of minutes and only make a couple of mistakes. The estimate is 1 to 3 mistakes per cell division. That's based on measured rates, and it's based on the known error rate of the DNA polymerases. They're not perfect. And plus, you get things like ionizing radiation hitting your cells and causing a DNA breakage, and the DNA repair enzyme has to fix it, and sometimes it has to make an educated guess, and it doesn't always guess properly. But that's not the reproductive cells. The reproductive cells are tucked away on the inside, kept away from sunlight and other things on purpose. And interesting, tomorrow I'm going to give a talk on coral reefs. Corals do not sequester their germline cells. They don't have selected cells that are held in the side, and so it's a big wonder how corals have persisted all these years without mutating out of existence. But anyway, sir. Yes, there are corrective things that occur in us that correct mutations. But once a mutation occurs, the body doesn't necessarily know how to fix it to the original. It can replace something that's broken if it's broken on one strand, but if a strand breakage occurs, it's not necessarily a perfect alignment. There are things where you can lose a part of one of the bases, and then when a copying occurs, it thinks it's a different base and replaces it with the wrong one. So yes, DNA replication is very accurate, but it is not perfect, and we know that. In fact, in part of my research when I was making those fluorescent fish, those weren't the original genes I pulled out of the corals. I mutated them. And the way I mutated them is I ran a PCR reaction with very bad conditions on purpose so that the polymerases would make mistakes. And when they made mistakes, I took that DNA, I put them into bacteria, I spread them out on a plate, and I looked for the brightest colony. And I selected for brighter colonies by intentionally inducing mistakes that were caused by the DNA polymerases. So DNA replication is accurate but not perfect. Hence, variation. Sir?
What amount of errors are still maximally possible so that you have a stable genetic population?
What amount of errors are possible to still have a stable population? I have no idea. In one of my other talks, I'm going to conclude that each person in the world has between 15 and 20,000 mutations that we've accumulated since Adam and Eve, and yet we're still alive. When we look at the human genome, the evolutionists for years said, oh, it's just junk, all that redundancy, all that duplication, whatever. Actually, the redundancy and duplication in the genome is the reason why you're alive right now. Because God engineered feedback loops and fail-safe mechanisms and duplication so that if something breaks, something else can take over and keep you alive. Okay, one or two more. Ma'am?
Can you address the apparent contradiction between punctuated equilibrium and gradualism?
Can I address the apparent contradiction between punctuated equilibrium and gradualism? I suppose I could give about a 3-hour lecture on it. You want a quick answer? Darwin's theory depended upon slow and gradual changes. We don't find slow and gradual changes in the fossil record, however. That's what Gould pointed out very nicely. And so therefore Gould came up with punctuated equilibrium, where he says that species have long periods of time of stasis, and maybe on the periphery, evolution might occur. And therefore you don't see it in the fossil record, but then later on, this species is taken over, and so you had a very rapid change. There's not, of course, there's been a war about this. And since Gould has died, punctuated equilibrium has pretty much faded away. I have not heard much about it over the last decade. But it actually addresses the fossil record better than Darwin's theory does. Darwin's theory depended upon innumerable transitionals, fossils, which we haven't found. And genetically, it depends upon innumerable small-scale changes in genes. And instead of changes upward, all we see is debilitating changes downward. And Sanford's going to talk about that in his genetic entropy talk. We don't know how species can persist for millions of years, let alone increase in complexity or evolve upwards. Okay, last question, way in the back.
Okay, given the huge benefits with livestock line breeding and breeding for specific maybe maternal traits or weight gaining traits, that sort of thing. Given the prevalency of line breeding in livestock and all sorts of other animal populations, why do the evolutionists and the humanists stay away from that? Because there's an inherent conclusion built into that, isn't there? Because if mammal populations are subject to the benefits of line breeding, why are human beings off limit?
That was a very long question.
I know the answer as a Christian.
Okay, I'm sorry. I tried to follow it. Can you say it in one sentence? Given the known benefits of selective breeding, why? Okay, why?
Why is that? Why are human beings not analogous to the same beliefs, strategies.
Okay. Why are human beings not analogous to the same strategies? Actually, I think we are, but there are moral problems with selective breeding in humans.
Well, then that's what I'm saying. I know the answer from a Christian and a moral perspective.
Oh, okay. Why aren't...
But that should be off limits to the evolutionist.
Oh, no, it's not off limits to the evolutionist. You've heard of eugenics. You've heard of Darwin's cousin, Francis Galton. You've heard the phrase that the eugenics was basically a Darwin family business, with his sons and grandsons being involved and other relatives. You've heard of the forced sterilizations of people. You have heard of possibly the Namibian genocide during World War I under the Kaiser, and then, of course, the Hitler's genocide against other peoples, the Christians and the Jews, especially the Jews, but Christians also, in Germany. Selective breeding is very much a part of evolutionary history, theoretical history and practical applications of evolutionary theory. They don't like to talk about it, but even in recent years, every once in a while it pops up again, and it's really a really scary proposition. And now, of course, we have all these genetic technologies that are coming online where people can select the sex of their child, selectively get rid of one if they don't like it. But also, we might have the ability in the future to select traits that code for intelligence or longevity and get rid of other people. Actually, I think it's always been in the background. It really, after World War II, the world was so had such a distaste for these ideas that it went underground, but it never really went away. I mean, geneticists today are lament. In fact, one guy, he was saying that given all the mutation right now, and the fact that we have all modern medicine that keeps us alive, despite the fact that half of us should be dead already, he says, but in the third world countries, that's where natural selection is going to be operating, getting rid of all the bad mutation. Actually, the hope for the future of humanity is in third world countries. The Western world is going to die because of mutation accumulation. So it's still there. It's just underground. Anyway, wow. That could be. I know CMI speakers have given talks on that subject, and that could be a long discussion just in itself. But eugenics is still there, breeding is still there, and boy, so much to say, and I can't. So, thank you all very much. Let's say a quick prayer, and we'll be dismissed. Father, thank you so much for your word. Thank you so much for new life. Thank you for salvation. Thank you for Jesus Christ. Lord, thank you for the world that you've created for us to live in and enjoy. Thank you for science, that we get to explore your world and rethink your thoughts and discover the wonderful things and the amazing things that you've done. Lord, help us to be diligent in pursuing. Help us to be diligent in applying. Lord, help us to be faithful in sharing our faith about you with other people. Amen.
We are live from Muskoka Bible Center. Rod Hember here, live on Bible Discovery TV. Just a quick note to Chris Ucia. Chris Ucia, who is our master control operator back in Orangeville, Ontario there. We're going to just stay live because in less than 15 minutes, we have Dr. Safferty coming up, who is going to be talking about the incarnation based on science. Now, there is a couple of different things we want to mention as we go on as well. Tonight, we have Dr. Danny Faulkner, Universe by Design. That's going to happen approximately 7 o'clock. And so it's a lot of excitement going on here. And by the way, if you're watching, I just learned that this particular signal is being pumped through the entire camp here at Muskoka Bible Center. If you're watching from your trailer, thank you for watching, but you're missing a lot of the action here. You can actually meet the scientist and be a part of it. Also, another quick note, we are still working on the internet connection that will allow us to put the chat room up and gather questions from online live. We are still running into some difficulty with that, only because of some of the connections where we're at are not as good. And all of our bandwidth connections are being used to bring you and broadcast to you the best possible picture we can from this remote location near Huntsville, Ontario at Muskoka Bible Center. Also want to remind you that Dr. Carter has an amazing DVD that you can find at creation.com, where he talks a lot about this. Now, just to review in the last hour, what we've talked about is genetically proving that it's impossible for the human genome to be longer than, a million years or so on, longer than 6,000 years. It's a demonstration that the idea of race is absolutely absurd scientifically. Of course, the Bible always said that. Another interesting point here is the land bridge, which Dr. Carter mentioned, across the continents from the southern tip of Africa all the way through to the southern tip of South America. The mistake we often make is we judge the past based on what we see in front of us now. And the world is changing, the earth is changing. And another point that Dr. Carter made, which I thought was absolutely fascinating, is that we are not evolving, rather we are devolving, and we can show that and demonstrate that by looking at human genetics. So this was an amazing session. Of course, Dr. Carter has other sessions coming up throughout the week, and in fact his next session will be on, I believe it is Wednesday, multi-dimensional genome. Okay, I'm not sure what that means, but I guess beyond the 3 dimensions of, or the 4 dimensions of time, space, width, and breadth. So multi-dimensional genomes by Dr. Carter, that's coming up on Wednesday, the morning session, approximately 10:30. Now coming up in just a moment, Dr. Safferty is going to be dealing with the incarnation from a scientific point of view. That's happening in about a little bit less than 10 minutes here. And so we're going to keep the live stream active and going. Another point I want to make here is that we have been supplied by equipment from Roland Canada, and I'd like to thank Roland Canada for being a part of supplying the equipment we're using. Roland Canada, of course, we have one of the most amazing switchers by Roland, the 440 switcher. It's an HD and an SD switcher. So if you're a production artist and you are watching this, I encourage you to go to roland.ca and check out Roland Canada for your great equipment needs, your greatest equipment needs. They certainly can help you there. Thank you to John DeVries for helping us do that. Now, also, we want to mention Bible Discovery TV. We will be putting up the VOD, video on demand sessions of what you're seeing over the course of the next few days. So we want to ask you to be patient with us, and especially to our wonderful friends down under in Australia. I realize it's the other side of the clock, and so things are very late for you. So we're working on that and crunching those particular files as fast as we possibly can in the midst of all of our production. Our production team is 8 people here in a gigantic RV called the Hurricane. So there you go. It's a great name for an RV, the Hurricane. All right, coming up, Dr. Safferty. It's going to be interesting, and I've heard his name pronounced like 8 different ways, so I'll ask him in a moment how to pronounce his name exactly. And also, we're going to be asking the speakers to repeat the questions so that you can hear some of the question and answer going on. Now, coming up a little bit later on, we have, of course, tonight, remember that, Danny Faulkner, great astronomer. You're going to want to be a part of that. So right now, we're going to go to standby for a few moments. We'll be back in just a moment to set up the session with Dr. Jonathan Safferty, and we'll talk to him a little bit, and then I'll bring you the prelude to that in just a moment. I'm Rod Hembry. We are live at Muskoka Bible Center here in Southern Ontario. Beautiful place. I want to encourage you to stay tuned. We have lots more coming. This is BibleDiscoveryTV.com. Stay there. We'll be back in a moment.